Okay, hi everybody. This is my ReproRehab final report presentation. So just to sum up some of the key things that I've learned while I've been part of the ReproRehab program, honestly, a lot of it, starting from the very beginning, is about making intentional decisions about setting up file structures for data management and data storage. A lot of the labs that I've been trained in have been longer standing labs with a lot of rich data that haven't always been set up ideally for use in data analysis. And so a lot of time that I've spent kind of fighting against that has been to reformat a lot of the databases and file structures that have been set up and so that they're usable by things like R, which is what I learned here in ReproRehab. And so for me, I am transitioning from postdoc to faculty position in the next year. And so making some of these intentional decisions are going to be really big as I set up my lab, and thinking about reproducibility and longevity of my data has been a huge learning point with ReproRehab. I've also, I really started with the basics with R, so I learned how to import CSVs and how to merge files to create a master spreadsheet. And I learned how to use R to view, clean, and analyze data, and also how to find and use appropriate online resources to really continue my growth in R, because I see this as a starting point for myself and my project is ongoing. And I know that I'll want to use R as a primary analytical tool in the future, so I'd really love to continue to grow my skill set in this area as well. In terms of accomplishments during the fellowship time, I've learned basic coding skills. This started with how to read and write the language, what the actual landscape of the program looks like, and then how to break down and look up the things that I don't understand. So this part was really big for me. I think it's hard to debug code, and that's always what I've struggled with, and I feel a lot more confident in how to do that now after my time during this fellowship. My project was to create a machine learning model using a lot of National Registry data for an F32 project that I'm doing. And so part of that was really part of my skill set I wanted to learn was how to import many sheets of data, how to organize these sheets of data, how to combine them, and how to clean them. And this took a lot more time and effort than I really ever anticipated it would. I should have known that. But really, I focused a lot of my time on cleaning these data sheets and using different data visualization techniques to make checks along the way and ensure that I was doing this in a reproducible and robust way. And then also assessing missingness of the data for the modeling that we were going to do. And then merging the data sheets for analysis and running some preliminary analyses. So right now we are doing model selection. I still have a ways to go with this, but I have been able to perform some preliminary analyses for this project as well. I have a planned abstract to submit for the combined sections meeting of the American Physical Therapy Association. That's due in July of this year, and then a planned full manuscript for the fall of this year as well. And I will definitely be sure to cite the ReproRehab program for that. And lastly, I just want to say thank you very much to our fearless leaders in Pod 2, Tanner and Andrew, the Pod 2 learners, and the whole ReproRehab community. This really has been an amazing process. I've recommended it to a handful of my coworkers here at Ohio State, and I look forward to seeing where the program continues to grow. So thanks so much for the experience.
Hi, my name is Kim Bassendale, and I am the Assistant Director of the Neuromotor Control Lab at Marquette University, and I am a learner in the ReproRehab program, and this is my final presentation. When I look back at the work that I was doing when I started ReproRehab and compared it to what I'm doing now, I feel really proud of my progress. One example just to share here is how many steps I would have to do with some of my data processing when I first began the program. And because of the program now, it's so much easier and it's so much reproducible, and I can really just visually see the results on a day-to-day basis. I feel that my technical skills in MATLAB have significantly improved during this fellowship, and also my ability to understand and to write code has gotten so much better, and I can see this in how much better my R code has gotten actually, even though I was focused on MATLAB skills in this fellowship. So my ability to take what I've learned in MATLAB and apply it to R, even though they're slightly different languages, has really demonstrated to me that I have a deeper understanding of coding, which is what I really wanted to get from this program. So not only has the reproducibility in my data management and data analysis improved, but also in the development of study protocols and the dissemination of our work. Working in MATLAB used to feel maybe 20% exciting and 80% daunting, but now I would say that this has really flipped to maybe now it's more 90% exciting and 10% daunting. Um, and maybe it'll feel like that forever, but I know now that I have more direction into what my goal should be for improving these skills, for tackling more challenging problems on my own. And really, I feel that I have joined a really positive community where I can reach out to others who are also excited about learning things and teaching others about things that we have learned. So back to what Sukli said, I do think this has been a very successful Ponzi scheme. During this fellowship, I have significantly improved the reproducibility of my data processing and analysis, as I had mentioned, and this can be used for a lot of future studies, and it can really go a lot of different directions from here. I have used what I've learned from ReproRehab to present at APTA CSM this past February. My goal for the program was to create a rough draft of a manuscript to present for one of our studies, the user experiences with our wearable systems. I'm still working on this, probably will be for the next month or two, but I have a lot of the results done, the figures, a lot of that, the heavy things I need MATLAB for, it's all done. So it's really just getting my background, my discussion section, those things done. In addition, I have also been outlining a second manuscript that I'd like to write, which is talking about a new method for looking at adherence to wearing our system. And really, I couldn't look at any of these methods if it weren't for ReproRehab. Maybe I would have, but it would have taken me far longer and it wouldn't have been as productive and as reproducible. And I've also shared a lot of the information that I have learned through this program with my lab with really positive results. And as I mentioned on the previous slides, working on those two manuscripts, and I'm hoping to, in the near future, to continue to work on different study proposals and different grants that I can also refer back to ReproRehab for support. Lastly, I just wanted to give a big thank you to ReproRehab for truly an amazing experience. And thank you also to all of my fellow students who are learners for creating a welcoming environment. Thank you to our pod leaders with a special shout out to Devin and Jin. And thank you to the administrators for everything you have done to make this possible and to continue to improve this program. I'm excited to see where things go from here. Thank you again. Bye.
Hi, I'm Sydney Bodendistel. I'm currently a postdoctoral research scholar at Washington University in St. Louis in the program of physical therapy. And today I'm gonna just review how I felt about the ReproRehab program, what I've learned, and then the paper I actually submitted this week. So for the first question, what did I learn in ReproRehab was basically the knowledge and competence to just use R more regularly. I used to do almost everything in Excel by hand, especially a lot of the data manipulation. So moving cells around or deleting cells or making alterations. Definitely figured out through ReproRehab that doing all of this in Excel is not going to be easier. It's not faster, and it's definitely more prone to mistakes. And so now with the project that I just submitted and worked on during ReproRehab, I do almost everything in R, and I feel really confident doing it. And even just more recently, we actually updated the data set that I was given, and that was a breeze through the use of R. I also used plots and graphs in Excel, and now I do almost everything in R again because it's just so much simpler, and I can always go back and make a minor change in just one line and get the same beautiful plot that I always wanted. In my pod, which was pod 3, we also talked a lot about some of the options that you can work with through R. So we had a lesson on R Markdown, which I'd never heard of before, but is super convenient. We did an intro to Google Colab and kind of walked through some of the features that option has. And then through my own learning time, I looked more into GitHub as a way to more easily share projects with people with little to no R experience. And so that I can put my code out there and not worry about people not knowing where to find it or how to read the code itself. So what did I accomplish during my fellowship time was I actually finished a project. It wasn't the same project that I had originally written in for, but it was using the same data set. So this data set has more than 300 people in it with multiple time points that are not evenly distributed and has hundreds of variables, most of which I did not use in this project. And so the data organization and data management side of the project was super heavy. And I don't think I could have done it without the use of R. And now I'm super confident in the research that I have and finished with this project. A really great example is recently this week, I got an email from one of my co-authors saying, hey, the variable I sent you is technically incorrect. We did a minor coding error. Here's the correct variables in a separate spreadsheet. If I was using Excel, this would have taken me hours, if not days, to complete. But using R, I timed myself, and it took me under 20 minutes to import the new data, replace it, and have the same old spreadsheet that I had been working on the whole time. I used to use SPSS for all of my stats, and for this project, I did it in both SPSS and R, and I got the same results. So that really gave me the confidence moving forward that if I needed to, or if I wanted to, I could switch all the way to R and not worry about having to have an SPSS license anymore, which is really convenient. And in addition, if I get reviews back that says I need to run something different, I have the code the way that I ran it originally, and I don't need to track those files down in SPSS. And when I need to run it again, it's so much easier. It's a click of a button because it's all right there in the code. And then finally, as I kind of mentioned earlier, I was able to learn how to use plots. Plots in R are so much easier than plots in Excel. They give you a ton more options that Excel will never have. And this is one of the plots in the paper we just submitted. So the box plots are actually from R itself, and then the information around the outside of the box plots help tell the story a little bit better. And so I was able to really easily use jitter and changing the colors and the fonts to get it the perfect size and tell the story that I wanted to with this data. Do I have any papers or grants planned for the upcoming year? Well, I just submitted the paper that you just saw the plot for, which is titled Differential Gait Features Across Parkinson's Disease Clinical Subtypes, and that's in the Journal of Parkinson's Disease. I definitely included the ReproRehab in the acknowledgement sections, including the grant number, because the time that I spent in ReproRehab definitely helped me complete this project.
All right. Hi, everybody. Thanks for watching my video. My name is Grace Bellinger. I use she, her pronouns. I am a learner in Pod 3. I'm currently a postdoc at Johns Hopkins School of Medicine. What have I learned in ReproRehab? A lot. To keep it short, I'm focusing on just a few of the most important things. So definitely R programming. I've learned a lot throughout this program, especially from Allison, one of my Pod 3 TAs. She gave a great overview of one of the core packages, and I've also learned about a lot of obscure packages that I might not otherwise have learned about from some of the other TAs in work blocks and people sharing things in the Slack channel. So that's been very useful for me. I also learned a lot of best practices in terms of data storage and code commenting for reproducibility. And I've been applying this both in work from my PhD lab that I'm handing over to colleagues there, and the projects I'm starting up in my postdoc lab, which I joined less than a year ago. I also really enjoyed learning about best practices in data sharing, since it's now required as part of a lot of NIH funding mechanisms. That was a new content area for me, so all those resources will be great to look back on when I get to that point. And I also really appreciated Joanna's overview of GitHub and have aspirations to really build up my GitHub repository once I wrap up some of these projects and start disseminating results. what I have accomplished, again, a lot through the skills developed through ReproRehab. One of my big accomplishments is just the resourcefulness that I've developed throughout this program. I think I've developed a toolkit, not only knowing specific resources to use, but just resilience and looking for resources online, especially with Stack Overflow and different programming tips. That also has built up my confidence. I think I really, through this program, began to identify as more of a data scientist and have a lot more confidence in my programming abilities and just my ability to navigate any challenges that I face when programming and analyzing large data sets. As far as actual deliverables, I submitted an ASNR abstract back in January, which I am presenting next week, which focuses on large data. It is from a smartphone application that can be used for remote monitoring of stroke outcomes. So lots of big data there in challenging format to use as far as how clean the data are. So I've been spending a lot of time in R processing that data and working on visualizing it. So if you're at ASNR, come by my poster. I also was will be the senior author on a conference abstract submitted prior to the all hands meeting. And I'm really fully responsible for manipulating, cleaning, wrangling, and analyzing the EHR data in that project. So it was really exciting to be a senior author and really take full responsibility for the data, which is looking at swallowing dysfunction post-stroke and different treatments for that, as well as outcomes. And lastly, a project that I've been spending a lot of my time on is this Fuglemeyer project, which I've taken to many office hours. Some of you may have heard about it. And I am planning to submit it for CSM in July to take there next February. This is an extremely large data set, so it's from our repository here at Hopkins that uses a battery of stroke assessments, Fuglemeyer being one of them. And we have this really rich data source, not thousands, 1,400 upper extremity Fuglemeyer scores. But that's a lot. That's a lot to work with. And it really allows us to look at impairment across the continuum of rehabilitation and how folks recover. So I'm interested in using these data to kind of phenotype those that do recover more than the minimal clinically important difference and those that do not. So those were all conference submissions and will eventually be papers as well. And grants that are ongoing are planned. So I submitted my AHA grant shortly before beginning ReproRehab. It was funded. And so any scholarly products from that grant will definitely mention ReproRehab. It's funded through the end of next year. I'll also have a manuscript on the Fuglemeyer project, just describe that figure is from, as well as the feasibility work I'm taking to ASNR and that dysphagia project, which was submitted to ASHA. All of these are expected to go to pretty high impact in our field. So like stroke, something about that level. And then lastly, at some point, I'm planning to submit a K99 to NICHD either October or February of next year. So that is in the works, and ReproRehab will really set me up for success in those ventures.
Hi, I'm Amelia Kane. I am a learner in Pod 1. I'm a physical therapist and a first-year PhD student at the University of Southern California. Because I'm a first-year PhD student, I think a lot of the things that I've learned in ReproRehab are more basics and kind of like intro skills, especially even just the first in-person meeting where we had introductions to like what is reproducible research and what are some of the basics in how to conduct reproducible research. That was really helpful for me. I've also learned a lot about just basics of MATLAB coding, especially considering that I don't know, when this ReproRehab program first started, I had very, very limited experience. I had probably downloaded MATLAB for the first time maybe like 2 months before this started. So I've made a lot of progress since then for sure. Also, just basics of data organization. This does not mean that my data is now all organized. This is very much a work in progress, like all of these things, but I think just having some general understanding of how to better organize my data and why that is necessary. Also, intro to GitHub. I was super scared of GitHub whenever I first met it, and I'm slightly less scared of it now, due really mostly to ReproRehab. I think most importantly, I have learned that there's a really solid community of rehabilitation researchers that are all committed to conducting good science and supporting one another in that process. Also very important, I have learned some introduction to programming music and introduction to data science memes. I certainly still don't understand all of the data science memes, but I understand a lot more than when I first started. So here's a little work meme for you. During my fellowship time, I have focused on conducting a secondary analysis to examine the relationship between overall gait asymmetry and metabolic cost of walking after stroke. To do this, I cleaned and organized kinematic and metabolic data for over 20 stroke participants. I then calculated metabolic cost of transport, step length asymmetry, and a combined gait asymmetry metric for each participant. Did data visualizations for individual and group level data, and then conducted a preliminary analysis using just simple linear regressions. So, little plug, if you want to hear more about the results of that, please come to Poster 71 at the ASNR annual meeting. I'm going to be in Poster Session 1 from 6 to 8 p.m. and would love to talk to you more about it and show you what I've been working on throughout ReproRehab. If you won't be there, then, and you're still interested in learning about this, I'd be happy to talk to you, not at ASNR, but spoiler alert, overall gait asymmetry is associated with metabolic cost of walking in individuals with chronic stroke, based on what we're seeing so far. Any papers or grants planned? Hopefully this will be coming soon. So the work that I've done so far is going to be part of a larger analysis that I'm planning to submit for publication within the next year. More importantly, I think I'm going to be relying on the skills that I've learned throughout my future research career. So thanks, ReproRehab.
My name is Si Wei. Today I'm going to present the final report for ReproRehab program. A little bit about myself. I'm a prior postdoctoral research associate at Washington University OT department. Currently, I'm doing clinical work as a home health occupational therapist. Okay. Next page. I learned a lot from ReproRehab programs, but here I listed a few things that are the most important for me. I learned the principles and the best practice for conducting reproducible research and fair data principles. And I also learned the idea of open science and how to do data sharing. The last but not the least is about R coding. So I learned a lot of basic R coding skills, how to troubleshoot an error, and how to find reliable R learning resources. For this program, my end product will be a manuscript that I'm going to publish. So during the program, I finished data wrangling and data analysis for my manuscript. And I also have written 1/3 of the manuscript. And here are a few screenshots just to show what I've done. But I'll jump to this page. Those are the actual articles I've been reading. So for this one, it's all about data wrangling. How do I convert variables into, the variables that I can do the analysis with. And also here, this one is the actual data analysis. So in the beginning, I've done some missing data analysis, and going down here, calculate sample size and power, perform descriptive analysis, correlation analysis, and here is about the end, that's the hierarchical regression analysis. And I also conduct regression diagnostics. All right. Okay. And so I plan to publish this manuscript. And this is a study to examine the association between leisure, social activity participation, and loneliness in people aging with long-term physical disabilities. And I used a data set that consisted of about 350 participants. And this data set was from a longitudinal data set, but I only used one of the, a single year, so it will be a cross-sectional study. And like I mentioned earlier, a hierarchical regression model was done and regression diagnostics was done. And now I'm writing the manuscript. And that's about my progress and my finals. And I plan on continuously doing writing the manuscript until it is done. Thank you so much. And I really love this program. Bye.
Hi, everyone. For those of you who I haven't met yet in ReproRehab, my name is Brooke Dexheimer, and I'm sharing my final report, which I've titled Successes, Failures, and Tears Shed. So what have you learned in ReproRehab? So before this, excuse me, and during my PhD, I learned to work with data using JUMP. That includes data cleaning, analysis, visualization, all of that. And so if you haven't used JUMP before, it's really user-friendly. I think it's a lot of point and click, and you can kind of embed formulas and columns in a very intuitive way. And it's great for beginners, but what I've found is that it was really, really challenging for me to keep track of my most current data set, which JUMP table was my most recent one. If I identified a bad trial or like a faulty data point and removed it, then I would lose track of which table is correct. And so then I had kind of this mess of, I was constantly double, triple, quadruple checking to make sure that things were consistent and that I was calculating things in a meticulous way. And I used a little bit of code, as you can see in the screenshot, to try to make things reproducible, but then I still really struggled with organization, and I always was really worried that I was forgetting steps and quite disorganized. And so what it ended up doing is costing me just so much time in terms of constantly checking my data and keeping track of the steps that I was doing. And I would try to take notes or have a lab notebook, but then I would lose track of all of my Word documents that were my lab notebook. It's been kind of a mess. And it worked okay, but as my data has gotten much, much more complex, I have realized that this is not a sustainable solution. And so during ReproRehab, I focused on learning kind of just general principles of data reproducibility, better organization of data. I worked with RStudio. I used MATLAB more as well. I joke that I broke up with JUMP. And so this is just, I took a screenshot from our lab laptop where I was running more complex analyses, and it saved me a lot of time in terms of keeping track of what exact steps I had done to clean data, what exact steps I did for analysis, the filter that I applied, things like that. And so it's been a really great, it's been like a frustrating learning process, but it's been a really great long-term and big picture. And it saved me more time on the back end. A little bit of upfront time learning R and learning MATLAB, but all in all, a good experience. And so I put on the slide here, after less bad, I say, because I just want to mention that I still have a long ways to go in terms of, because what we've talked about some in ReproRehab is data reproducibility far beyond when a project ends, right? Or when it's published. And that's still something I really, really want to focus on, is making sure that anyone can reproduce my findings, that I can pass my data to someone if they need it. I can have an organized system of keeping track of data even after I published the corresponding manuscripts, things like that. And so that's still been a work in progress for me. But at this point, I felt like I've made great strides in just like within a project, really having this nice running list of every single thing that I've done with the data and how I'm able to reproduce that as needed. So a success, I would say, but still much to learn. So what have you accomplished during your fellowship time? I have 2 data sets that I really feel like ReproRehab has contributed a lot to, or my time participating in ReproRehab has contributed a lot to. And I had really grand plans during this program to work with MRI data and work with database I had access to, but of course in academia, everything takes 10 times longer than we think, and everything moves much slower. So, but I still feel like I got a lot out of this program. The square on the left here this is what I'll be presenting at ASNR. And so I won't talk too much about it. We also presented at the American Occupational Therapy Association annual meeting, but we're just looking at a specific brain region and its role in motor representations. And then also we have a precision drawing task on a tablet. And so I analyzed it in a different way and used R for that. And I feel like it really, really helped to make sure everything was clean and done correctly. And then I also used some of this preliminary data on a pending KO1 I submitted in February. So come to my poster ASNR if you want to see. The cube on the right here is a different set of data that I have been using MATLAB and R during my ReproRehab time to clean and analyze. This was data collected at my old institution. This was not collected during my ReproRehab time, but has been analyzed during this time. And it's that same brain region we're looking at: motor adaptation, gross motor reaching movements, and then again, motor representations. And so the manuscript is in prep for this now, and so I'm analyzing the data in a different way now. And we also use some of this data and put in R01 the end of last year. And so I'm co-I on that. And then we have a pending my pending KO1. It's the same one. I didn't put it to KO1. It's the same one. I just used preliminary data from both for different aims. All right. I'm over time here, so I will wrap this up quickly. Do you have any papers or grants planned for the upcoming year that use what you worked on in ReproRehab? Yes, we'll do the 2 manuscripts from the data I just talked about, 2 or more. And then we have a funded project through Shirley Ryan, and it's in individuals with Parkinson's disease, looking at lateralized presentations of tremor and bradykinesia, and then also lateralized functions of the basal ganglia. So I'm going to use some of the code that I've created during ReproRehab it'll very nicely allow me to analyze this data pretty easily. And so we're in the process of collecting data for that, and then any manuscripts result from that, and then it's a pilot grant. So we'll plan to submit a larger follow-up grant once we wrap this up. So thank you very much. Feel free to email me if you have any questions, if you want to chat, if you have any further advice for me on how to break up with JUMP, I'm happy to hear it. A quick plug, my department, which is the VCU Department of Occupational Therapy, we have an open faculty position. You do not need to be an OT for this. So anyone doing rehab research, if you're interested, reach out to me, or you can find the position listing online. Just wanted to plug that really quick. But anyways, thank you so much. And for those of you attending ASNR, I will see you there. Bye.
Hello, I'm Shamli Dusanne. I'm a postdoctoral fellow in the physical therapy department at Northwestern University. And this was my first time trying to systematically learn coding, specifically MATLAB coding, since it has been used in our lab very frequently. Through ReproRehab, I was introduced to several concepts for the first time, got to learn about GitHub. And since we work in a team, this is very useful skill to kind of make changes and to contribute towards our lab projects. I also got to learn more about the MATLAB coding fundamentals, which to the depth that I can now use them to write my first code. I was able to use some of these learned skills to compute basic gate parameters and also make some beautiful plots using different functions that I learned. I think I got better at troubleshooting the errors and trying to interpret the existing code in my lab. I think I'm more confident in the basic understanding, and also have better access to resources and to figure out solving complex issues. It was really good to know about the best practices during coding, and I also received lots of resources from the TAs. A big shout out to both the TAs. For helping me understand and get through the initial inertia and understanding these concepts. During my fellowship time, I was able to analyze some of the pilot data from stroke, and we were trying to answer a research question to see the effect of movement amplification environment in people with stroke, and this was walking data. I was able to compute some basic gait parameters and also make some graphs. Going forward, I plan to look into more details to see what are the sensitive measures that can assess this change. And I think the overall takeaways from ReproRehab is that I have more confidence and more access to resources. And I also have a group that I can rely on to ask my questions at all times. For now, we don't have any plans to use this, but definitely the bigger picture is to use this pilot data to write a K99 award for the postdoc, and I hope I see that coming through. Thank you so much to the organizing team of ReproRehab for making this possible. I really appreciate all your efforts. Thank you.
Hey, everybody. Excited to be here. Hopefully we'll see you all, or some of you, at ASNR. So I'm just going to do a brief presentation about my experience in ReproRehab. So I started out as an old dog here. I've been trying to teach myself how to code in R for a little bit, but really did not have much of an idea of what I was doing. So that prompted me, after a colleague told me about ReproRehab, to apply, and fortunately was accepted. So thank you to everyone. And here's what I've learned so far. So I've really learned a lot more about data science, and particularly about reproducibility. Previously, as a researcher, I focused on trying to make my research methods reproducible in terms of how I collected data, but didn't really think about the data analysis and data management piece and making that reproducible. So I had always thought of R as a way to help me visualize and analyze my data, but not really thinking about how to make that data management and analysis portion reproducible. So I think I gained some foundational reproducible skills in R to upload, clean, organize, and munge the data to prepare for data analysis, and develop some basic visualization analysis coding skills in R that are also reproducible. Also, just of interest, here's some other resources I found. I found a data management site at Harvard has some really good tools and other information about data management, so you might want to check it out. So what have I accomplished? So as I learned all these reproducible skills and coding skills, I created about 50 vignettes to help me. We have a large data set of an ongoing cohort study examining the prevalence and impact of non-obstructive sleep apnea sleep disorders in people with stroke. So I created about 50 vignettes that we pull in the data from REDCap automatically, but it needs to be cleaned and organized because it's being collected long and wide across 4 different time points. And so being able to take that, clean it, merge data, so then you can actually use it for the analysis in an appropriate way. I guess specifically, in addition to those 50 vignettes, I created kind of 3 main projects that I've been working on during this time here, using some of these skills. The first 2, however, I have to admit that I've been a part and I've been helping with it, but I haven't been the lead. Some of these skills are still a little bit beyond me, but I've been fortunate to work with some good colleagues, both here where I'm at Emory, and a colleague who I used to work with at Clarkson University in upstate New York. So the first one was a data fusion, so combining self-report log, activity monitor data, and GPS data to merge it and fuse it together to explore mobility in the home and community in people with stroke. And I'll be presenting that, some examples of that at ASNR in a poster. And the second one is survivor curve analysis, the life of a walk. So kind of unique way to look at patterns of walking that hasn't really been done in the literature that we can find. Most of the time, at least in the stroke literature, walking activity is being looked at kind of using summary data, like the mean number of steps across some time that they wear it, the activity monitor. So here we're looking at durations of walks and those patterns using survivor curve analysis, and that we're going to present at World Congress of Neurorehabilitation in May in Vancouver. And then the other major project I kind of started working on is looking and using compositional data analysis to explore the impact of sleep, activity behaviors, and sedentary behaviors after stroke. And so code analysis kind of, again, another unique way of looking at these activity data. So instead of looking at each individually, it looks at a composition of these three behaviors and how does that impact recovery after stroke. So I'll do a quick example that kind of combines some of those. Here's some of the code I wrote that actually preps all the data from about 6 different data sets, not data sets, but 6 different instruments from R, and cleans it and gets it all set to do the compositional data analysis. So you just click that button, boom, does it every single time. It's amazing. And then now it's just creates a quick table, looks at some of the summary data of some of the different measures that we're using in this compositional data analysis. And then I won't go through the whole CODA analysis, but one thing that you can create as part of the compositional data analysis is a ternary plot. So it's again looking at combining sleep, sedentary behavior, and active behavior and looking that together. So I just saved a file, but also created a plot, interactive plotly. So here you can kind of scan over these different subjects and see, for example, this person here spent 77% of their time in sedentary, only about 4% in active behavior, and about 20% of their time in sleep. So again, just looking here, this person had the most, looks like, activity. So about 35% of the day was either standing or walking, 29% was sitting, and about 36% was in sleep. So it's kind of nice that you can just push a button and get all that done. And so here's some of the papers and grants that we have been using it with. So previously, back in January and February, presented some of these data from this sleep and stroke study that we have going on at the International Stroke Conference and at the Combined Sections meeting back in February. Coming up, we're going to present the data on the fusion approach, combining that data to look at mobility in the home and community at ASNR, as I mentioned before, also at World Congress of Neurorehabilitation. The code analysis that I mentioned, here's another example of that ternary plot, using that for our renewal of our RO1, and also actually been using some of the skills I learned here to examine our DPT program outcomes. So now I have some code written that I can just, like magic, push a button and compare our DPT programs across years in terms of admissions and student outcomes. And so thank you to everybody, Lee and Keith, for guiding me a little bit, especially Tanner and Andrew, that were successful in helping an old dog learn new tricks. And really excited to see everybody at CSM and future meetings for continuing my journey in coding in R. So thank you and had a really great time.
Hello, my name is Devin Austin, and I am one of two TAs for the 2023 cohort of ReproRehab, along with one other TA for Pod 1. So what have I learned in ReproRehab? While I'm not a learner, I'm a TA, I still learned several things, both about how I like to teach Pod 1 and what Pod 1 has taught me. The first is that I found it challenging to teach programming to several individuals who have various different skill levels at once. It really forced me to acknowledge different perspectives of the same concept I was teaching in order to effectively get my messages across to everyone and to keep people engaged. The next was that I have taught programming before, but I've never taught programming with an emphasis on reproducible programming methods, and I found that I actually really enjoyed a lot, especially seeing those methods come across later in more of the self-learning portion. And one of the ways we did that was, if I take us to the ReproRehab GitHub, during the teaching portion within Pod 1, we had several weeks of several different MATLAB assignments. And within each week, we had individuals upload their assignments to GitHub and really play around with it to get a bit of a light-side learning experience every week and to continuously build on that. And lastly, my expertise primarily revolves around upper limb kinematics. However, it was really fun to learn to see some of the learners' expertise in real time. I was able to learn about some more lower limb kinematics, wearable sensors, and EMG data. So the big thing that I accomplished during my time in ReproRehab was this TA project, where I worked with another TA, Duncan Tulamary, to produce 2 videos on data visualization in both MATLAB and Python. And this was really to show people some of the ways that data visualization is different in MATLAB and Python, what's more convenient in each programming language, and some of these smaller things that we do to help make figures for presentations, posters, and papers. So we essentially start with a drawing, which is what we typically do in our lab, of what we want the figure to look like. And in the video, we go through how to recreate these figures and add a little bit of fancy pizzazz in both MATLAB and Python. And this can all be found on the ReproRehab GitHub, and the videos will be uploaded to YouTube. And the code for it for both MATLAB and Python can be found under TA projects for Devin and Duncan. And we have the live scripts that are made within the videos for both Python and MATLAB, as well as 2 master scripts that we have made beforehand, which include all the same information. And lastly, thank you.
Hi, everyone. My name is Christina Hall, and I am a PhD student at the University of Southern California and a proud member of Pod 1. And this is my final report presentation. So this year, I feel like I've come such a long way as someone with no coding experience. I was a bit nervous initially, but everyone's been so patient and gracious with me. And I have Jen and Devin to thank for giving us some formal instruction, both in GitHub and MATLAB. In GitHub, it was something that we had been using in the lab, but I didn't really understand the differences between like cloning and forking and branching, or how to use this to collaborate and share our work. So that was really helpful. We also did activities that had us use the fundamentals of MATLAB in sort of a different way than we did in class, which I think was really good to give you more exposure and practice. I think the overall main thing that I can take away from ReproRehab is just not to be afraid of the data or the code, and to have the confidence that whatever the next step is with processing my data, that I can do it and I can figure that out. And I think that's pretty powerful. Oh, and I learned that I can also relate to some of these programming memes that I always seem to come across on Reddit. So you can see here, I actually got bingo. I feel like I've accomplished a lot during my fellowship time. My project involves investigating whether individuals post-stroke can use a visual biofeedback that is informing them of their step lengths relative to neurotypical adults at different gait speeds. And a lot of the fall, I was trying to understand the purpose of each line of code of this pre-existing code that I had inherited so that I could adapt my project to it. And once this code started to make more sense to me, I was able to do some data cleaning and reorganizing in a way that allowed me to calculate things like step length and step length asymmetry, as well as distance from the targets or the error. So these are some of the examples from an example subject. Next steps for me are to bin the last 30 strides of each trial and use the average step lengths during that period to calculate error from the neurotypical targets. And then I plan to apply a linear mixed effects model and see what we can learn from that. Unfortunately, I have not yet submitted any papers or grants, but I do plan to submit abstracts to both SFN and CSM. And I do see a F31 application in my future. So thank you all so much for having me. And I really appreciate being in this program, and see you all at ASNR.
Hi everyone, my name is Dr. Andrew Hyman. This is my final report for ReproRehab in the year of 2023 to 2024. I was a TA for Pod 2, and this is what I have learned over my time here. So what have I learned in ReproRehab? Well, I kind of took this question more in his perspective as that of a TA rather than that as of a learner, because I don't know, I feel like there's a lot more impact that can be had in reproducibility if you can really try to teach these things in a kind of better way, given the context and the kind of limitations that we have within this type of format, being remote and kind of meeting somewhat infrequently. But the kind of main thing that I think I've learned from my time being a TA this year was really to try and assume more of a top-down approach when teaching coding, and really being able to just have exercises that people can kind of play around with and kind of see how they can just kind of change things here and there, and it can have this whole big different result, and that can kind of, I think, is a way to be able to present coding in a way that's more fun rather than a bottom-up approach, which is like, well, this is a data structure, this is an algorithm, this is a for loop, and those types of things are very kind of dry, somewhat very important, but I don't think they really kind of kickstart people's kind of enthusiasm for learning to code as they're juggling everything else in their academic life. Better understanding of creating a curriculum as well. I think with my co-TA Tanner for Pod 2, we did a really good job of really trying to create a curriculum early on that we thought would be very impactful and interesting for our learners given their interests. And so we really tried to develop something that was, I think, in a very short amount of time that could be considered very useful and reproducible for a lot of the learners who may not have any experience with coding in R, which was the coding language that we used. And then really try to, again, tap into a learner's curiosity more than anything else about how coding can really be leveraged and utilized, and really trying to present many different ways in which there could be some excitement generated about really trying to pursue learning to code rather than just trying to learn, how to do regression in R versus, not doing it in SPSS anymore. That type of thing is important as well, but I don't think it really creates this kind of long-term learning enthusiasm that you need if you're really going to take on learning a whole new programming language. So what have I accomplished during my time this year as a returning TA? I really believe that I was able to do a better job of being able to help sustain the ReproRehab community. I think there's like a lot of staying power in being able to really know that these ideas of reproducibility, this learning how to code, these are all things that we all struggle with and are dynamic and interesting problems, but if we have people that we can work on and rely on to work on them together, I think that's a super important, impactful thing that can really be taken beyond the program as well. And being more deliberate in making my own code and data more organized, I think there's a lot to be learned in terms of how with everybody's own research studies, how are you going to write code for this specific type of person who may want to use a specific type of way, and how should that code then be organized in a way that allows it to be just taken and no questions asked. They can just kind of run with it themselves, and you never have to meet the person. Hopefully they just cite you and it just becomes this wonderful thing. So really trying to be more deliberate about how to do that is really something I've been thinking about more than just trying to code quickly and create a result and not necessarily have it be reproducible. And then really adopt a learner's focus on iteratively reframing reproducibility as more technology is always added into mix. So really trying to continue to think about how to be better and better at being reproducible. And that's about it. Psych, actually I have this really cool graph I want to show you. So this is a graph that I made of the data that I'm going to be presenting at ASNR, and these are people who in red are the baseline where they lived, and then in blue this is where they live now, and then these arrows show where they moved to between the 1 year. I'm really interested in using these kind of idea of how moving can impact your motor skill. If you move to a better area or a worse area, how does that really impact overall if you are able to retain, or if just the act of moving actually causes some kind of stress that would actually cause you to not retain motor skill as well. And overall there were about 41 people out of the 349 that I looked at in this 1 year study, and pretty much people stayed within the median kind of income within the zip code that they started at until they went to. So that was kind of interesting, but basically people weren't moving on up, and people weren't moving on down. So for the most part they basically stayed even, which was kind of interesting. And do I have any papers or grants that are upcoming? I will probably be citing ReproRehab until the day I retire or die, whichever one comes first. So yeah, there's a lot coming. I'm very slow because I'm trying to be reproducible, but I will be citing it quite a bit. Thanks everyone.
Hello, I'm Audrey Kellerman and I'm a learner in Pod 3. I've learned a lot in ReproRehab, mostly about using R, its capabilities, and better practices for reproducibility. So this has included R Markdown, GitHub and VS Code, and Google Colab. I've become much more familiar with R and coding language in general, and asking questions. So now I can ask people or websites ways to approach wherever I'm stuck. And I know enough to interpret answers or modify them to work for what I need. And errors don't stop me nearly as often as they used to. So through these experiences, I've also learned reproducible practices for data cleaning, analysis, and creating figures and tables. Before ReproRehab, I would frequently export to Excel or SPSS for some steps if I wasn't sure how to complete an R, but I'm happy to report I've learned enough that it's now much easier and better practice to perform all steps in R with everything documented. And this has been exponentially simpler when rerunning, when we have more participants to add or making changes based on co-author or reviewer suggestions. And during my fellowship time, I've completed a lot of ReproRehab videos and tutorials on ReproDB2, mostly about R. And that was more at the beginning stages of the fellowship period, and later as needed based on different stages or tasks required of different individual projects. So the self-directed nature of the program was very well suited to my learning and changing needs over the course of the year. And I submitted one paper and and then the subsequent revisions to it using skills learned in ReproRehab. And I also prepared 2 aims of my dissertation using our skills learned here, from the beginning, merging files, cleaning data, analyzing, and generating tables and figures. And I defended my dissertation and now preparing to submit those 2 aims specifically to journals as well. And then I started a side project examining resting state functional connectivity and falls using ReproRehab resources, from understanding what I'm looking at with that kind of data and how to handle it. And as that project will be ongoing, I'm grateful that the resources will be available after my time as an active learner ends. And as I mentioned, the one has been submitted and it's under revision, and I cited ReproRehab for that. The 2 that are part of my dissertation will be submitted within the next couple of weeks. And the resting state one is a little farther off, but it's definitely started in using skills here. So all of these will cite ReproRehab. So thank you so much for a wonderful year and teaching me so much. And I'm excited to continue to be a part in whatever capacity and have access to the resources and get other people excited about joining and learning from this wonderful program.
Hi, I'm Morgan Kelly. And in ReproRehab, I got to learn a lot about best practices for data analysis and data sharing. In Pod 1, I gained a lot of useful skills using MATLAB to help with conducting reproducible research. And this included data cleaning, data visualization, and statistics. I also was able to get a lot more comfortable with GitHub, specifically using it as a version control and a data sharing tool. During my fellowship year, I got to work on a couple of different projects going on in our lab. I work in the gait rehabilitation and motor learning lab at USC, along with 2 other ReproRehab learners. So this first project, Cognition as a Moderator of Motor Learning Post-Stroke, with Amelia Kane, we spent some time improving the data analysis pipeline for this project, which is an ongoing trial. And then on my own, I worked on coding a biofeedback task to help study motivation in chronic stroke. And this was part of this 3rd study, post-stroke apathy and mobility. So the biofeedback task that I was working on is a task that's actually been used before in Parkinson's disease, but I was not able to access the original task, so I had to recreate it from scratch. This task requires real-time signal processing, visualization, and analysis, so there's a lot of new things I hadn't done before. There's 2 parts to it. The first part is an instructive motivation task, where you're shown how much force you're being asked to exert on a pinch grip sensor. And then you see a scale with the goal force exertion marked on it. And you squeeze the pinch grip sensor until you reach the goal. And then it tells you if you've met your goal or not. The second type of task in this biofeedback task is an incentive motivation task. So instead of telling you how much force to generate, it'll show you how much money is at stake for this trial. And then you see that amount of money on a grid and you just squeeze as hard as you can to try to win as much of that money as you can. This grid is scaled so that your MVC, your maximal voluntary contraction, is actually around 50%. So in this example, there's 50 euros at stake. The person looks like they're squeezing to about 22 euros, and their MVC, their maximal, is probably around 25. And then they get some feedback about how they've done. So I had some hardware that I had not worked with before that I got to learn a lot about. We had a pinch grip sensor, a FUTech panel mount display, which has both analog and digital output. But after talking with some people in ReproRehab and at my lab, I ended up actually wiring it through a DAC board, which helped get the input into MATLAB a little bit easier. So I ended up doing all the data manipulation in MATLAB. For the software, this also was really new to me, like trying to figure out how to code this biofeedback task. But I talked to a lot of people and I had some help and actually got it working. I still have some tweaking to do, but I think it's looking pretty good. So for the instructive trials, it'll show you how much force to generate, it'll show you the scale, and it'll show you in real time how much you're actually exerting. And then it'll give you feedback about whether or not you've hit your goal. And for the incentive task, it will show you how much money is at stake, and you can see in real time how hard you're pinching, and then we'll show you how much money you've won. So this was really fun and also pretty challenging for me. I started off trying to use Psych Toolbox in MATLAB, but just didn't know a lot of people who knew a lot about Psych Toolbox and was kind of struggling with that software. So I ended up getting some advice to just do it in regular MATLAB, pulling up like a window and setting everything to a timer to get this to work. So I've piloted a couple of participants and we're going to start using it in stroke survivors soon. So for that first project, fixing up our data analysis pipeline in this cognition and stroke, we had a poster at ASNR, and then once I can get my task working for chronic stroke survivors, I'm going to use that data for my dissertation and hopefully for publication. And then those of you who met me at the kickoff event may have noticed I was pregnant, so the last thing that I've accomplished during my fellowship time is the birth of my daughter Gwyn, who already has been a rehabilitation research participant. This is her participating in SPEEDY and intervention trial.
Hello everyone, I'm Cynthia Lee. I'm associate professor at the University of Texas Medical Branch, and here is my ReproRehab final report. I have learned three things, major things in ReproRehab. The first one is the R programming skills. So I learned the basic R programming, including how to installing packages, importing data, clean data. I know it all sounds so basic, but I feel this is a very great program that allow me to really have enough time to really use it and really have very hands-on experiences. Also, the data wrangling. This is kind of the part that takes the most of my time, kind of just trying to figure out how to really make sure the data is ready for any analysis. And it takes, I think it probably, just like all the other statistic analysis, that you probably spend 70-80% of your time managing your data, and you spend 20-30% of your time to analyze the data. And this is kind of how I feel. And with the support from the TA and the pub, the word block, and that really helped me to have the time to really be able to learn how to manage the data. I also learned how to do some simple data analysis, also how to generate tables and plots, and how to convert the output to Word document. I think the most important thing I learned is to actually be able to problem solve when there's any programming issue occur. And sometimes it does take some patience and also like discussion with the TA. The second thing I learned is the concept of reproducibility. I think this is like getting like more and more important, and I'm very glad to be able to learn this and be able to know there are lots of available sources out there like GitHub, and I think the transparency is so important. So other people can actually reproduce the result you generate. The last one, but not least, the networking. I think the pod, I'm in pod 2, and I think our TA is phenomenal, and all the work I have been participating, the TA also have been amazing. I think it's great to have a group of people who have the same goal to really learn the skills together. And I think ReproRehab really provide a very wonderful platform for everybody to be able to engage in this networking. opportunities. I have gained a lot of familiarity of using the R programming, and so I'm able to really use some of them, not all of them. Kind of like just underground practicing because I'm actually a SAS user, so it's really easier for me to just go ahead to use SAS. But I do spend some time just using R and try to see if I can generate the same result as SAS, and so it's kind of nice. Right now I have a Learn Scholar project, and I have the electronic health records, and it's a great place for me to practice what I learned from this program. I don't have any papers or grants like publish or submit yet, but I do plan to do so. And particularly for the project, I actually try to practice the programming of the R using the R programming. So that will be definitely in my future. I will be more than glad to include the ReproRehab grant number in any of the papers or grant in the future. Thank you.
Presentation for ReproRehab. I worked on two main projects. Well, one main project and one sort of secondary project as part of my time in ReproRehab. My main project was a longitudinal study of median nerve movement in the carpal tunnel. And my second project was part of my dissertation, which was a social network analysis of hand therapy research co-authorship. So what have I learned in ReproRehab? First, during the first phase of the program, we learned about general principles of reproducible research and data management. For me, the most important aspects of that were precise procedural documentation. So keeping track of all of the methods that I've used precisely so that it can be replicated by somebody else later, especially for my main project, which may be taken up by someone else in the future. Second, machine readable file and variable names, which informed kind of how I constructed my dissertation project. And also careful annotation of code within the code I was writing for both my main project and my dissertation project. Two, I learned a lot about different R tools and packages. I was sort of a novice in R coming into this program, so I feel like I've greatly developed my skills communicating code and results to lay people through R Markdown, data management with tidyverse, sort of the packages within tidyverse, and then also regular expression for text data visualization with ggplot for my longitudinal data, and then iGraph for my social network analysis. And then also just general computational logic for formatting and operationalization of variables. Next, I learned a lot about integrating external tools into coding, such as ChatGPT or Gemini sort of AI tools for problem-solving coding solutions, and then also being able to communicate with the AI to improve the solutions. Additionally, I used a lot of Stack Overflow and had to learn to sort of adapt and repurpose the responses there and sort of Frankenstein different approaches from different solutions. My main project was my longitudinal study of the median nerve movement. This project ended up requiring a lot more data management than I originally anticipated. I was hoping to spend most of my time on analysis, as I think always happens when we start a new project, and most of my time ended up being with the data itself before analysis. I did a lot of scrubbing, so identifying and correcting errors, which I did not know were supposed to be cleaned beforehand, but was not. Management of the data, that is updating variable names to actually make sense. Splitting and merging data from different sources, and then also subsetting the data I had to do analyses from different groups within our population. Next, I did a lot of data manipulation, transformation, absolute values from a distance, a lot of distance data, and then normalization of data to kind of be able to compare it. I did some exploratory visualization, which unfortunately didn't show any significant trends over time in our population. So technically, maybe it was fruitless to then go on and do statistical analyses, but I did them anyway, mostly for my own learning experience using mixed modeling. Future plans for this project, mostly combining what we have with another related data set and kind of creating maybe an omnibus sort of 360 view of this issue and or talk more about the procedures of collecting that data. So here's just a few snippets of the data visualization that I did. We also used other things like spaghetti plots and whatnot. My second project was a social network analysis, like I mentioned, of hand therapy co-authorship. So things that have been published in the main hand therapy journals is what we're starting with. I also had to do a lot of data scrubbing, management, and manipulation with this particular data set because it was inconsistently formatted. There was a lot of text data from different sources, and it was a very large data set, so it took quite a while to problem solve handling it. It was almost 2,000 authors and over 5,000 total connections. I was able to finally get a preliminary network visualization with a graph, and my future plans are to add author attributes to this visualization, do sort of subgroup visualization, and kind of explore more of the structure of the network through statistical approaches used in social network analysis. Here is my very rudimentary first attempt at visualizing this network. Any future plans? I will be submitting scientific abstracts to ASHT, the hand therapy annual meeting for our national organization, and then also through to the international organization, which has a triennial conference. Both of those deadlines are coming up soon. I'm also going to be continuing on with these projects so we can eventually submit them for publication in musculoskeletal rehab-related journals, such as the Journal of Hand Therapy. And then obviously my dissertation project will be published within my dissertation as well. Lastly, I just wanted to thank everybody who worked so hard to put this program together. And I wanted to thank my fellow podmates in Pod 3 and our TAs for really helping me to grow a lot in my coding journey and just being able to manage and manipulate and analyze data. Thanks, everybody.
Hi, everyone. My name is Kirsten McCartney, and this is my final report presentation for the 2023-2024 ReproRehab cohort. I was a learner in Pod 4. So throughout ReproRehab, I've learned the basics of data science and data reproducibility. I've learned how to conduct object-oriented programming in Python, including kind of how to run tests for my code to make sure that it's running what we think it's running. And I've also learned how to simulate data in Python. I have created and successfully used GitHub for a project with a colleague and kind of have gotten in the process of continually updating this every time I'm kind of working on a new branch. So that's been a great new skill to have. I submitted an abstract for the 2024 ASNR annual meeting, and I'll be presenting the poster next week. And I've developed code in Python, which has aided in the successful completion of the analyses related to my first and second dissertation aims. I do currently have one paper in development that's based off of aim 1 of my dissertation, and I have, at least for right now, an additional 2 papers, which I will use what I worked on in ReproRehab, and I may submit a fellowship application to the AHA in the fall. And I've really enjoyed my time. Thanks, everyone.
Hi, everyone. My name is Michelle, and this is my final report presentation for ReproRehab. I am a Pod 3 TA, and I primarily know how to use Python. I'm currently a PhD student at McMaster University. So, I've learned quite a few technical and soft skills in my time at ReproRehab. The one I'm most excited about is the amount of development and progress I've made with my R skills. So again, primarily I've used Python in the past, but because everyone in my pod essentially was using R, most of the troubleshooting and the content was R focused, and so I find myself a lot more comfortable using R for my own research as well. Better communication for coding challenges. So I was working with a couple peers, like one-on-one, just trying to help them navigate their own code, whether that be looking at like specific examples of functions and trying to figure out what each line was doing, or maybe trying to further understand the logic so that they can reproduce the same function or the same kind of idea in R. And this really helped me with my understanding of programming and what each function was doing, and just like better understanding like the syntax and the logic behind problem solving. Which brings us to problem solving skills. So I think that kind of goes hand in hand with the previous point, but I essentially think that I got better at Googling the issues that I was having and becoming more efficient at finding solutions for them. I think especially from the kickoff event, I did learn a lot about good data science and reproducibility practices that I'm very excited to take back into my own research and bring to my own lab. And finally, and I'll be going over this a little bit more in the next slide, but I learned a little bit more about VS Code as a coding platform that I'm very excited to share in my lecture that I created as a TA. So during this time, I explored VS Code further as a text editor, as well as creating an intro to Python using VS Code tutorial that will be linked to the ReproRehab page soon. So using this, I was able to find a lot of perks of actually integrating VS Code in my current coding practices, and I found it a lot more streamlined to work with my prof and with our collaborators on the same project using VS Code. Additionally, I have become a little bit better at knowledge dissemination skills. So just training the current mentees that I have in the lab with programming projects that they currently are undertaking. And it's also, ReproRehab has really helped me reflect on my own pipelines, my own practices, to make sure that everything I'm currently doing and will be implementing moving forward is reproducible and transparent and just really clean and well structured. So currently I don't have any manuscripts or grants that I'm working on, but I am going to be teaching a prerequisite for a machine learning for the physiologist course that my supervisor teaches. So in this prerequisite, I'm hoping to be going over a lot of the similar content points that I did for the resource that I created for ReproRehab with regards to the crash course in Python using VS Code. And because a lot of my peers have created or identified really helpful R skills as well, I'm excited to be bringing both that and Python content to students who will be taking the machine learning course. I'm also currently working with some undergrads that are interested in learning more about programming, so that's something that I will continue to teach using some of the resources I've gained through ReproRehab, so I'm very excited about that.
All right. Hi, everyone. My name is Allison Miller. I'm a postdoctoral researcher at Washington University, and I was also a TA for Pod 3 this ReproRehab cycle. And so, well, I've learned a lot in ReproRehab, but for the purposes of this short video talk, I'll focus on kind of two key things. The first is that I learned a lot of really helpful, new and useful tools that could help streamline my personal and collaborative workflows. And so the way that we organized things in Pod 3 is we had different members of the pod share a technique or a skill or a tool that they use in their workflow that they thought others might find helpful. And so, for example, we had one of our pod members, Amanda, do a really great tutorial using Google Colab. We had another one of our pod members, Audrey, share with everyone how to use our Markdown. And then Michelle, my fellow TA, shared with everybody how to use VS Code. And so these were tools that I had heard of in some shape or form, but really hadn't had a good idea in terms of how they worked or how I could use them. And so after these great tutorials, I feel like I have a better sense of how I might be able to leverage these tools in some of the work that I'm doing now in some future projects. And in particular, I think a lot of these tools could be really helpful when working collaboratively with others. The second key thing that I learned that I, again, didn't have much knowledge or experience with before ReproRehab was important concepts related to harmonizing and sharing data. And so I had, not had a lot of experience with sharing data before, and I certainly hadn't taken on a project really where I'd harmonized data. And so I've learned a lot of really useful skills through both the ReproRehab didactic content and in embarking on my own project where I had to do these things. And so what have I accomplished during my fellowship? We have finally finished harmonizing over 10 years of accelerometry data that's been collected by members of my lab and collaborators. And we're preparing now to submit the data to NICHD-, which is their data and specimen hub. And so the submission is pretty much ready to go. We're just waiting on a couple of institutional signatures. And so my pro tip would be, if you are sharing data and you need those signatures, give yourself some extra time. And we are hoping that this data set, depending on how long it takes for Dash to review things, we're hoping it'll be available there in the coming weeks. So I'm really excited about that. Lastly, I have a few papers or grants planned for the upcoming year in which I will acknowledge ReproRehab funding. The first is a poster that I will be presenting at ASNR this year on the harmonized data set. So I look forward to seeing anybody who's attending that conference then. I'm also preparing a manuscript to submit to Scientific Data about the harmonized data set, in which I plan to acknowledge the ReproRehab funding. And then I'm starting to pivot towards preparing a K99R00 submission, in which I plan to reference the harmonized data set that I've worked on and some of the tools and skills that I've learned in ReproRehab. So a lot of upcoming things for the year. And thanks so much for listening to this, and take care.
Hi, my name is Alex Nilis, and this is my final report presentation for ReproRehab. I was a part of Pod 1, led by Jen and Devin, and our focus was on MATLAB. My fellowship with ReproRehab was invaluable for me as I go into my dissertation. One of the things that I really appreciated about ReproRehab is that it's training us in a skill that we don't necessarily get through our program. Some classes are sometimes offered on like data analysis and coding and things like that, but that's just not something that was available to me at the time here at UNC. So the ability to do it through ReproRehab was really, really helpful. My experiences prior to ReproRehab can be very succinctly summed up by the Michael Scott quote. And I knew exactly what to do, but in a much more real sense, I had no idea what to do. Because I had done programming before, like I'd taken classes in Java and C++ and things like that, but it's just different than processing data. So like I kind of understood, like I understood how code worked, but I just didn't know how to really go about it, and especially in MATLAB, which is so useful. But it was just kind of new to me, and I didn't really know where to start. I kind of lacked the confidence. So the ability to be a part of Pod 1 with a focus on MATLAB with Jen and Devin was really, really helpful. Other things that I really appreciated about ReproRehab was just the introduction to GitHub. I kind of knew what GitHub was, like I'd had like a statistics professor host his textbook on GitHub before, but I didn't really know what a great resource it was, so just having that introduction to GitHub was really helpful. And then also the introduction to reproducibility and data sharing was also really helpful. I submitted an abstract with data from our lab group that was collected before my time. The people who collected it weren't here anymore, and it was very tricky to kind of figure out how things were processed and what some of the variables were. But then I also got a data set to do the power analysis for my dissertation, and everything was so clearly labeled. It was so helpful. So just that example, being able to tie it back to what I learned in ReproRehab about how important data sharing and data management and things like that are to save time, and then also just make things more accessible was really important and really helpful. So as I mentioned, during my fellowship for ReproRehab, I developed the code for analyzing the gait biomechanics for my dissertation. I'd hoped to also get the code to process the EMG data that I also plan on collecting. I didn't quite get there yet, but I will still be using those skills from ReproRehab to build in that code as well. So I have all of the stuff for the treadmill gait biomechanics that I'm going to do. So a quick 10-second summary of my dissertation is I'm going to see how a visual disruptive task changes gait biomechanics in people with ACL reconstruction, and I'm also going to compare that to a cognitive task to see if their gait changes in a way that could predispose them to post-traumatic osteoarthritis. So that's what I'm going to use the code that I developed for. In order to develop this code, I used my skills that I learned in Pod 1 and then also completed 2 courses in Coursera that I found via the ReproRehab website, which was really helpful. They were from Vanderbilt. One was an introduction to programming with MATLAB. The other was an introduction to data signal and image analysis. I should probably put an asterisk next to completed that one because I did skip some of the image analysis stuff, but either way, it was really, really helpful. Finally, I do have 3 manuscripts planned for my dissertation. Each will use the code I developed to analyze the gait biomechanics. Therefore, each of them will have ReproRehab as a funding source. Thank you for the opportunity to be a part of the program. I really enjoyed it and have highly recommended it to everybody that I can tell about it. Thank you.
Hello, my name is Jin. I was a TA of Part 1, and here's a presentation of my final report. What have I learned in ReproRehab? I learned 2 things as a TA. My focus was on designing activities that may help novice MATLAB coders the most. While many of Part 1 learners were certainly not novice coders, some were less skilled than others. The first thing I learned was that I should help learners practicing building loops, such as for loops or while loops. People all knew about the concept. The implementation of the knowledge, however, needed more work. Another thing I learned was that people are much interested in using custom functions. Often coders need to assemble MATLAB's basic functions to design a custom function, performing more complex tasks. Again, people were mostly cognizant of the mechanisms of a function, be it custom built or provided by MATLAB. They just needed more practice. So I prepared 2 tutorials on MATLAB functions. One is about writing one's own function and testing it. Here's a screenshot of the script I prepared. The task was to fill in the blanks and complete a function named myCrystalBall. The function takes a table and different arguments. Depending on the specific arguments, the function returns different answers. So there were 5 tasks, with each task having several subtasks. The tasks were all based on the features introduced during the boot camp period. The first task was the use of if-else statement. The second task was to use an input parser to identify what function arguments and their values were provided. The third task was defining a variable using a switch case statement. The fourth case I didn't include it here because it was just another use of if-else statement. And the last task was working on forming the right error message. Then I also prepared another tutorial on the details of functions in MATLAB. This was based on the requests of some learners in my pod. Hence, I prepared 4 short interactive notebooks on the definition of here 1, global versus local variables, how to handle errors, understanding workspaces, and types of functions. The notebooks were prepared with MATLAB's live script so that people can read paragraphs and run executable code chunks at the same time. Live script is a relatively new feature of MATLAB. For those experienced in Jupyter Notebook, you can consider this as the MATLAB version of that. Finally, anything to acknowledge ReproRehab's help? Not yet. I prepared an abstract with my lab mate and submitted it to ASNR, only to fail, unfortunately. However, I have some products I worked on when I was a learner in the program, and this is an extension of that I worked on during the last 2 months, in addition to the aforementioned TA project. This is a GUI that helps with reviewing wearable sensor data simultaneously with the video. I built this to verify if a wearable sensor signal pattern matches certain leg movements of infants lying supine. I can navigate frames by clicking buttons here. My lab does a lot of wearable sensor data analysis, and in associating sensor signals with movements, this tool I expect to be helpful. I use Python's PyQt to build this. If this becomes more official and public, I may be able to thank ReproRehab properly. Thank you, and that's the end of my presentation.
Hi, I'm Sam Randolph. I was in Pod 4 this semester, or this year, and this is my final report. So this year, my goal was to kind of transfer some of my skills that I had from programming in R to Python. And I feel like I really accomplished that, and that was like my main objective. So I'm pretty pleased with that. Really grateful to my TAs and also the other learners in the pod who were also learning Python with me. I feel like they were just so helpful, and there was a lot of resource and knowledge sharing going on, and I appreciate that. I also learned a lot about just data management, open science, GitHub. Thanks to my TA, Joanna, for her presentation on that. I feel like that was really helpful. And before Repro, GitHub was very overwhelming for me, and just it's not hard to use, but it feels like there's like a bit of a steeper learning curve. And Joanna really made it simple and easy, and I appreciate that. I also just in general learned a little bit more about debugging and using online resources and what it means to find documentation for certain packages. And so that's been a big help for me in my projects, both in and out of Repro. And then I learned the very basics of machine learning and AI, and specifically cluster analysis, which is what I was doing. During my time in ReproRehab, I've been working on a K-Modes cluster analysis project, which I designed and implemented from start to finish. So I used a Medicare data set, about 8 years worth of data. And I ended up having over 200,000 participants in my sample. And so that's the largest sample size I've ever had. And I wanted to create a typology of adults aging with depressive symptoms, since older adults tend to present a little bit differently with depression. And I found in my analysis that my sample was having a lot of cognitive and a lot of sleep problems. And so it kind of points to areas for future intervention and something to look out for in this population. I visualized my cluster analysis using 3D plotting, thanks to Duncan for walking me through that. And I am about to submit an abstract for this project for the Society for Research in Psychopathology annual meeting this fall. So yes, hopefully I will have a manuscript from my Repro project. The conference that I'm submitting my abstract to is in October, and then I, of course, would try to revise my project based on feedback, and then I would be submitting the manuscript. So thank you so much. I've really enjoyed my time in the program, and I'm grateful to all the TAs for hosting work blocks, and for my TAs in particular for making our pod so much fun, and so, such a great learning experience. So thank you.
Hi everyone, my name is Tanner Reese and I am a co-TA for Pod 2. And before I give my final report, I wanted to give a quick thanks to all the learners, all the TAs, and all of the members of the leadership team for helping make ReproRehab happen. It was a great program and a great few months I spent with you all, and I learned a lot from it. So to start, I had no idea really. I'd heard of GitHub before, but I didn't have any idea how to use it before ReproRehab. So I'm thankful now to at least be able to start using it for code sharing and project collaboration. And then a few of the things I focused on for my own research are basically how to get rid of loops from a lot of the functions I had written. Using this per package specifically, I found very helpful. And then also how to incorporate parallel processing into these functions to increase computational efficiency. And then finally, I wanted to better understand error handling for dealing with some of the singularity warnings and non-convergence issues we'd get for some of the models that we run pretty frequently in our work. And then finally, I wanted to really solidify my understanding of mixed effects models in motor unit research and basically how to better specify random effects that accurately reflect the study design to basically just model the data more accurately and to better address our research questions. So in RPod, the content I was sort of responsible for introduced the basics of data wrangling, data visualization, and statistical analyses in R. And then over the last 2 or 3 months or so, I kind of put some of the things I had been focusing on into writing a pretty well-functioning bootstrapping script that can perform non-parametric bootstrapping on multiple mixed effects models on some of the pretty common motor unit variables that we test for our work. And then I was able to incorporate aspects of that code into performing some multivariate analyses as well. So things like linear discriminant analyses and principal component analyses as well. I was able to write some code to help our lab and answer some of the research questions we're interested in. And in that vein, I was thankfully able to assist on some of the statistical methods sections that our lab is presently working on. So ReproRehab will be cited as a funding source on those. And then a recent project I had gotten involved in the last couple of weeks is this datathon that's happening here at Washington University in St. Louis. So we are essentially working with some fictitious electronic health record data and training a machine learning algorithm to predict 1-year mortality. And we're basically competing against other teams against or within WashU. So that's kind of been stretching my brain a little bit, but it's been good for me, and I've learned a lot from that process so far. So as I mentioned, our lab is utilizing some of that bootstrapping code that I had written, and is going to be utilized in a handful of manuscripts that we write. So ReproRehab will be cited as a funding source on those. And then in this next few months, I'm hopefully going to get off the ground. What I hope will be like a methods paper regarding proper specification of level 1 variance covariance structures for modeling time series data with mixed effects models. So I want to again thank everyone for making ReproRehab a very stimulating and fun experience for me, and I hope to see you all in the future.
Hi, I'm Lane. I was in Pod 1 for ReproRehab. Go Devin and Jen. For the work blocks and self-learning period, I mostly focused on an EEG analysis. I knew how to do it in EEG lab, but I did not know how to like code it myself. And it was just really time consuming to do it individually in EEG lab. So I used our for loop structure that Devin and Jen taught us to do that pre-processing, batch processing for EEG. Since then, I've done like event locked potentials, and then I'm moving into an event locked coherence analysis. The second one I'm a little less familiar with, but I'm starting to kind of put it together with some help from people here at UNC that do this pretty often. Other things I've done in MATLAB are load cell analysis. This one's pretty straightforward. The data is not too messy. It doesn't even have to be filtered. But in the code on the right, I did do a filter just because it seemed like the room we were in was adding a little bit of noise. And then also I've started working in R. There's a fun penguin data set that I've been messing with that has like their beak length and their like flapper length. So it's just kind of a fun way to learn. I like the data visualization a little bit more in R than I've been able to figure out in MATLAB. During the fellowship time, kind of everything I just said, it's also just been a really busy like dissertation time. So I resubmitted my F31, which is hopefully in review. And then my dissertation pre-proposal is passed and my proposal is coming up. So just a lot of stuff on the back end, and that it's been nice to have like the individual work blocks every week just to have that hour dedicated to something related to ReproRehab, which is a lot of my work in my dissertation and grant. For upcoming papers, the EEG event-related potential paper is the first one that's listed up top. That should hopefully be submitted by end of summer. Really excited about that because it's kind of a new analysis in sports medicine for emotional processing. Because who knew that knees have brains and humans attached to them. For the event-related coherence analysis, this one's kind of just a fun project between myself and another student here. He's more familiar with the coherence analysis, and then I'm bringing the event-related part to it. So we're going to tag team that and hopefully submit that by end of fall. The EMG profiles, that is kind of also a group effort, but that analysis or that processing should be fairly easy and straightforward. And then big thing on the horizon is just dissertation. And that is it. Thank you.
Hello, everyone. So this is my final report for this year's ReproRehab cohort. Yeah, what a year. I've learned a lot this year. And first of all, during the teaching phase, I learned to structure group lessons, communicate with the group, and give and receive feedback. This year's pod was way more advanced than last year's pod, so we had to adjust the material, which was also a new experience for me. I also prepared some lessons during this phase, in particular one on scikit-learn, and this increased my knowledge of the library, especially as the topic goes around clustering, which I've not worked that much before. During the self-learning phase, I focused on creating content for the ReproRehab YouTube channel, and beyond mastering the cringe of seeing myself on video, I obtained lesson-specific skills and also video editing skills. What I accomplished, I think, at least something during the fellowship. So we kept meeting with the group during the self-learning phase. Yeah, that was one accomplishment. And the other accomplishment is that I created 4 videos for the YouTube channel. They focus on running Jupyter notebooks in a Python environment. So that's 2 of them. The first one is on setting up the environment. The second one is an introduction to pandas in a Jupyter notebook that runs out of an environment. And the second 2 videos focus on creating a video version of the Git course. Yeah, and at the moment, I have no papers or grants planned for the upcoming year. Yeah, thank you.
Hello, my name is Anne Schwartz and I'm a postdoc at UCLA, and I've been a learner in Pod 3 under the guidance of Michelle and Allison. During the course of ReproRehab, I mainly learned to refine my data management skills in R. This included combining different data sets, as well as setting and manipulating variables inside R rather than outside of R, and using packages such as tidyverse to do so. One of the biggest takeaways from ReproRehab for me, I think, was to automatize and comment as much as possible when I'm running any sort of analysis, not only for efficiency reasons, but also for reproducibility and helping me to go back to a project after a little while without wondering too much of what I did there. Additionally, I learned to conduct cluster analysis in R by using packages such as factor extra, dent extend, or cluster grid. And I started working with Visual Studio Code for running R, Python, and or Bash scripts in one application rather than several different ones, which hopefully will increase my workflow and efficiency in the future. Finally, and additionally, throughout our weekly pod meetings, I further gained insights into tools such as Google Colab for code sharing or ChatGPT for troubleshooting some specific code lines, which has shown to be helpful. These skills and tools finally helped me to accomplish my two main goals for ReproRehab, of which the first and main goal was to finish the analysis on the relationship between brain imaging measures and changes in arm impairment after stroke, which was a secondary analysis of the VNS rehab trial. The second goal that I accomplished was that I could finish the cluster analysis for characterizing stroke-related arm impairments based on items of a clinical scale in quite a time-efficient manner, and already shared it with the collaborators waiting for their feedback. And third and last but not least, I made my first steps in Python, which is hopefully just the beginning. Concerning publications related to my work in ReproRehab, I successfully submitted my main project on the association between neuroimaging predictors and changes in arm impairment that has been accepted for the ASNR conference as a poster and as a short boost talk, and the associated paper submission is in preparation and will also acknowledge ReproRehab. Finally, to conclude, I would like to thank the whole ReproRehab team and all of the members for creating this inspiring and encouraging environment that helped me build up my computational skills, and also always keep the motivation level slightly higher than my frustration level. Thank you.
Hello everyone, I'm Dr. Vikram Shanai Handiru, a research scientist at Kessler Foundation in New Jersey. I'm the director of Neuromuscular and Electrophysiology Lab in the Center for Mobility and Rehabilitation Engineering Research at Kessler. Our lab focuses on studying novel neural engineering tools and methods such as non-invasive brain stimulation and recording and smart wearable sensors for improving the motor functions in individuals with stroke and traumatic brain injury. I mainly joined this ReproRehab workshop to learn the best practices of open science and data sharing. And I'm a learner in the Pod 4 led by the TAs Duncan and Joanna. So my main motivation why I joined the ReproRehab workshop was to learn the best practices of data sharing with respect to the neuroimaging data. Especially after reading this paper on open and reproducible neuroimaging from study inception to publication, article on neuroimage. So what have I learned in this workshop? So during the boot camp, I learned various new tools relevant to efficient data management and file organization. And I learned the simulating the data for reproducible statistical analysis, and tools and packages helpful for making reproducible plots for manuscripts and presentations. And also fundamentals of GitHub. So Johanna taught us like how to create our first repository. I learned how to make like commit changes, pull requests, etc. And I also learned best practices for sharing the data on different data archiving platforms such as OpenNeuro, AddDep, and Dash. And some of them are recommended platforms for the grant funding agencies as well. And so during the fellowship time, like during the self-guided learning phase in the last 3 months, I learned the essentials of brain imaging data structure, or BEATS, which is the recommended standardized format for neuroimaging data, specifically like EEG and MRI. And I got to know, like, I got familiar with the example data sets, and I had like hands-on practice with them. And I also wrote scripts to programmatically set up the MRI and EEG data collected as part of my grant-funded pilot clinical trial study. And to give a like little bit context of my study design, we had this study funded in during the pandemic, and this is a double-blinded randomized control trial to compare the effects of active versus sham high-definition transcranial direct current stimulation, which is combined with Music Glove Exergaming, which looks like a Guitar Hero video game where participants are supposed to make these finger grips as they see these musical notes land on the screen, and the sensors pick these fingertips. And basically this is to improve the engagement during the repetitive motor practice. And this repetitive motor practice is combined with the neuromodulation using HTTDCS as an adjuvant treatment. And like every participant underwent 20 minutes of brain stimulation, followed by like 45 minutes of this exergaming for 10 sessions in the 3-week period. And as part of this study, 14 individuals completed the study, like all the visits, including baseline MRI scan, a baseline EEG session, and 10 sessions of this intervention, which is like combined TDCS and Music Glove, and 2 more EEG assessment sessions, like at the end of intervention and a 2-month follow-up. And so this is the like summary of the study design, and the main focus is to share the MRI and EEG data collected during like visit 2, 3, 14, and 15 on Open Neuro. That's the goal with which I started the self-guided learning phase. And since the data collection was concluded in like last year, December, it coincided with the self-guided learning phase. So, and the study was double-blinded, so we only removed the blinding in January. And since then, I have worked on standardizing the entire data set in BIDS compatible format. And I'm just awaiting the IRB's approval for sharing it on Open Neuro. So I plan to use some of the data visualization and dissemination tools that I learned during the workshop in my current manuscripts, which are under preparation. And I would like to acknowledge the support I received from our Pod 4 TAs and fellow learners, and also great work block hosts. Thank you.
My name is Molly Shepard. I was in Pod 4, and this is my final report presentation. So what did I learn in ReproRehab? The primary things I took away from ReproRehab were I really increased my Python skills. So our pod kind of covered a lot of the main packages that people use in Python, including SciPy, NumPy, Pandas, all of those kind of things. So I'm now kind of more comfortable with Python, and I've used it a little bit in my own research. Additionally, I learned some of the machine learning techniques. So some of the ones that we covered in our pod include, we did some neural networks, closest neighbor, nearest neighbor, naive Bayes. We looked at some decision trees as well as random forests. I haven't actually used any of these yet in my own research, but I do have some more kind of familiarity when I can discuss these things now and actually understand what they mean. Now, during my time in ReproRehab, I've been primarily working on my aim 3 of my dissertation. And this aim is looking at the influence of sport-specific activities, specifically running and hop cutting, on hip joint reaction forces in individuals with hip dysplasia. So in this work, I do a lot of 3-dimensional musculoskeletal modeling, and a lot of the steps in running these models are really time intensive. So I was running steps in these models that took at least 2 hours per person, and I'm running these on 40+ people frequently having to rerun things. So it ended up being like this multi-week process of trying to run these models, which is just not very good at all. So what I ended up doing was utilizing what's called the Open Science Grid, which basically you're able to submit things and the Open Science Grid kind of farms them out to different computational resources at different institutions across the US that are not being used. So you can submit a job and maybe I have 40 individuals that need to be run and each one will go to a different computer. So instead of being run in perpendicular and running one after another on one individual computer, they can all be run at once. So instead of taking 80 plus hours to run, they're all done in the span of a day. So I did that to run my models, and in order to submit to the Open Science Grid, I had to access the OpenSim Python API to kind of code what I wanted to do to my models. So this was my first time really using Python in my project, and then I submitted those Py files to the Open Science Grid. And I have some of my results kind of shown here. So these are different components of the joint reaction forces. So we have the anterior posterior, which is forward backward, superior inferior, which is up down, medial lateral, which is kind of in towards the middle of the body and out towards the outside, and the overall resultant components. And here in running data, we found that individuals with DDH had higher medial joint reaction forces than controls during the early portion of the gait cycle. And here's a little, this runs fun model of a person running. So I am planning on submitting this paper I've been working on to the American Journal of Sports Medicine. I'm hoping to finish that up by the end of the month. I have also submitted abstracts. I presented a poster at the Orthopedic Research Society annual meeting in February, and I cited ReproRehab on that. And I've also submitted an abstract to the American Society of Biomechanics meeting, which is happening this August. And that is also work that was supported by ReproRehab. So thank you so much. I really enjoyed being part of this program.
Hi everyone, my name is Tarun and I'm a PG student at the University of Texas. And I'm also a learner in Pod 4. And I'm excited to share what I have learned with ReproRehab. I had 3 goals when I joined the program. One is to understand and learn the principles of reproducible science, which included data standardization, management, and sharing. 2, to effectively use Git and GitHub for version control and open access code sharing. And 3, to network with ReproRehab fellows and learn about their research. In addition to that, I also wanted to learn how to simulate data and conduct a power analysis and build an analysis pipeline before collecting actual experimental data, and also to visualize data using other softwares like Matplotlib and Seaborn, as I've been using ggplot2 and MATLAB for a while. And I'm glad to say that I have achieved all my goals. I networked with the brilliant scientists here and learned about their research during the talks and interactive sessions. I implemented the data analysis and visualization principles that I learned during the bootcamp to my project. My project, where I analyzed behavioral and electrophysiological data, which was recorded before, during, and after participant learned a motor skill. I do not have the data here because it's unpublished data. And finally, as a side project, I also built a dashboard to track progress across multiple research projects and manage inventory for internal use in the lab. And a quick snapshot of that is here, and you can see that we have a lot of gauze and EEG gel. So my project was accepted to the Neural Control of Movement Conference as a poster. It's titled Repetition but Not Learning Related Corticospinal Tract Plasticity is Enhanced During Sensorimotor Mu Rhythm Trough Phases. If you plan to attend NCM, please swing by my poster. I'd be happy to chat. And this manuscript is close to completion, and I plan to submit it by the end of summer. Interestingly, we organized our Care Research Day a few weeks ago, where we had the opportunity to invite Dr. Teresa Cruz from NICHD as the keynote speaker. And it was really cool to see her feature ReproRehab and our group picture that we clicked at LA when we met for the meet and greet sessions. I'd like to take this opportunity to thank the organizing teams, the TAs, and my fellow learners for giving me an amazing learning experience. I plan to return to ReproRehab as a TA later this year, but otherwise, I'm always available on Slack and through email. If anyone wants to chat, I'd love to know more about what you do. Thank you so much.
My name is Lauren. I am a fourth-year PhD candidate at Washington University in St. Louis, and this is my final report. So for things that I learned during ReproRehab, I think the biggest thing I learned was a lot of new data reorganization techniques. The project that I worked on involved data that was collected from different sources. So some of it was sensor data, some of it was self-report measures, and some of it was assessments that performed with the patients. And so a big thing I learned here was how to get all of that data into one big CSV file to make sure everything was able to be processed in the way that we wanted to do that. The other thing I learned a lot about is GitHub. That was something that prior to starting ReproRehab I had heard of, but didn't really know how to use or how people used it. So that was something that I think was really helpful for me to better understand how other people use GitHub so that I can start to work that into my usage as well. And then the third thing that I learned during ReproRehab was really just good time management. So I personally found the work blocks to be really helpful to have that hour of dedicated time to work on things. So for me, that was one of the biggest benefits of having ReproRehab, was to say that I knew I would have this hour every week to be focused on this project, and knowing that I would have people there that I could help with asking questions and things like that. I think that was a really helpful part of ReproRehab for me. As far as things that I have accomplished during this, I was able to finish all of my data collection for the third aim of my dissertation, which was the project I was using for ReproRehab. I was able to do all of the analysis that we planned, and we were able to present those findings at the APTA CSM meeting back in February. So it's really helpful to have all of that get accomplished during this time period. As far as going forwards, I just submitted a paper for publication using this data set, and I'm sure over the next few months I will be working on revisions and things like that for this paper. At this moment, I don't have any plans to apply for grant funding, but I'm trying to figure out what my next steps are post-PhD. So once I figure that out, I will let you all know. And that is all I have.
Hello, my name is Duncan Toolmary, and I was a TA for Pod 4 during the 2023-2024 ReproRehab cohort. What did I learn? I learned that rehabilitation researchers are an extremely smart group, and I'm proud to be among them. I also learned that reproducible rehabilitation research will become common practice, and I'm excited to see this happen. What did I accomplish? I co-led Pod 4 with Joanna through bootcamp using integrated learning. I also helped 5 pod members with their self-guided learning projects. Some examples of these projects are machine learning for exercise dosing and group clustering, refactorization of code for future open source use, and learning and using Python for data analysis. Do I have any papers or grants planned for the upcoming year? No, I do not, but I do have jobs coming up in this next year. And so I plug ReproRehab for all my job applications. And finally, I just wanted to say thank you for everything, for another great year, and looking forward to seeing what everyone else has done. Thank you.
Hey everyone, this is Jeff Turner, and I'll be presenting on automating a movement screen for my project, representing Pod 4. The movement screen that I am aiming to automate is the landing error scoring system. This is a jump landing test that is often employed as a screening test. It's also a modifiable benchmark and a return to play or sport task. It is commonly manually scored, where the person would be video recording someone performing the jump landing, both in the frontal and sagittal planes, and then they mark off a binary score for different components that we will go through very briefly at the initial contact and at the end of the landing phase. My overall goal for this project was, again, to develop an automated scoring tool for that landing error scoring system. For this, we're going to be using OpenCap, which requires 2 smartphones. It's a free-to-use software, highly recommend. This software processes 3D coordinates and joint angles, which our tool, through Python, will read in those motion files for either individuals or for batches. It will provide the user quality control checks, and it'll provide, most importantly, the actual scores for this landing scoring system. Since everyone likes the ending first, these were what I would hope to be my accomplishments. The first is gaining an expertise in the use of OpenCap for markerless motion capture. The second is actually developing this tool in Python, and this tool would utilize me gaining experience in classes, functions, loops, modular functions, and then lastly, dictating this entire project in GitHub to maintain a level of 1, transparency, 2, accessibility, and then 3, reproducibility. This slide is a quick overview of what the landing error scoring system looks like. Essentially, it's having someone, usually an athlete, performing a jump landing from a box that's 30 centimeters high to a distance, 50% of their height from their location, and then having them perform a follow-up rebound jump. The less is operationally defined by 17 individual items. Each of these are on a binary or ordinal scale, and you can see the operational definitions here, but we won't go through them. Essentially, what our goal was to turn these operational definitions into modular functions within Python. So let's go through a quick example of what the jump landing looks like. And here we have our athlete performing the jump landing with our 2 smartphones using OpenCap. So before I show some code, here's just a bird's eye view of our steps of how I am writing the code to process our data and get a less total score. First, we have our OpenCap setup, followed by extracting the pose in kinematics, which is automatically done through that software. C is going through our key events detection. This is occurring within our Python script to determine when initial contact and toe-off is. If we have those time points, then we can actually extract those biomechanical features. Knee flexion at initial contact, hip flexion at max position, etc. And here's just one example of one function that we wrote to identify knee flexion errors at both the initial contact and max position time points. And we wrote one of these for every single component that we had. And fast forwarding to the end, if you hit run on the Python script for however many trials that you had, this is what you would end up with within Python. This is easily exportable spreadsheet that shows us our total score and all of our errors. Quickly to go over some preliminary results on the actual criterion validity, because one of my goals was to look at, well, we created this tool, how does it compare? And what we came out with were moderately high ICCs for comparing OpenLess to an expert rater. Additionally, OpenLess against the force plate for the actual timing of events came out to be exceptionally good. This was done in a 12-person sample, so I look forward to, in the future, performing this on a larger pool of individuals that we've already collected on. Now the next step is analysis. And so this is how far I've come along. We've built out how OpenCap interacts with our Python code, and we've had it all transparently located on GitHub. The next steps really for me is to look into building this into a nice GUI, which is a web-based user interface. Additionally, we'll be discussing these results at the upcoming ACSM conference. I'm also looking into a technical report write-up and publishing it soon. Lastly, I want to give a big shout out to my Pod 4, Duncan and Joanna, and the rest of our team. We really made an awesome pod. I really enjoyed our experience in the fall, and we wrapped it up really nicely with everything in the spring. Thank you so much.
Hi everyone, this is Ryan from Pod 4, and I'm a postdoctoral research associate at the University of Southern California. I focused on a few things in ReproRehab. So first and foremost, I wanted to develop RStudio data management skills and also to get better at writing clear, accessible, and reproducible RStudio files. And some specific applications of RStudio skills were applying multiple imputation techniques to improve survey data analyses. I work with a lot of complex survey data that gets messy, and in cross-sectional studies, sometimes missing data is a problem in particular. So I have some projects I was working on with cross-sectional data. And then two other more minor things I worked on were understanding latent class analysis to include a method section in a research strategy document for an upcoming grant proposal that I'm submitting. And I dove back into some of my dissertation work, and I looked at psychometric network analysis packages in RStudio to carry on with some analyses that I had started, but then froze for a while I was getting situated here at USC. And most importantly, too, it was great to develop relationships and network with other rehabilitation researchers. Programs like this are great because we have opportunities to meet with people whom we wouldn't otherwise meet. So it's been great hearing about what everyone's working on. And some things that I've accomplished during the fellowship time. So the first two entries are publications that went into the Journal of Diagnostic Medical Sonography. So the first publication applies multiple imputation techniques to a cross-sectional study that I've been working on with my mentor, Sean Roll. And then the second publication is an invited editorial that I offered to the journal to complement the first paper on top. And then I also had two conference presentations that I developed, poster presentations for the first publication. One of them was at an ergonomics conference that happened last week. And I've also been working with a post-professional OTD student named Tom Casey in the lab here. And it's the same general data set, but it's a different data frame that we've been working on. So applying some of the ReproRehab skills to mentoring him and working with Excel and working with SPSS has been a great opportunity as well. And then the last entry there is a presentation based on the grant proposal that I'm submitting, and that was just recently accepted. So I should be going to present that, and I leveraged some of the ReproRehab resources to learn latent class analysis in the completion and writing of the abstract and grant and proposal. And I have a few things so far that are planned for the upcoming year. So I have a longitudinal paper that's the big project, and I used some of the ReproRehab time to put together 4 different data frames from the same cohort that we've been tracking for a few years in the sonography workforce. And then the second paper is a full paper that I'm developing with the post-professional OTD student working in our lab with Tom May. And that should be also submitted to the Journal of Diagnostic Medical Sonography. And then I've been working on 2 grants. One of them, the first, will go in next week, so fingers crossed for that one. And I've been working with my PI and his collaborators on some of the methods sections and research strategy document in general with an overall R01 that they're hoping to submit based on the work with the sonography workforce data set. And that's all that I've got, and thank you so much.
So I broke down what I've learned with ReproRehab into broader buckets, looking at principles of reproducibility, specific technical skills, and confidence to apply these technical skills to make my own research more in line with best practices. So to demonstrate what I've learned over the last few months, I should probably show you where I started back in September. So first, looking at principles of reproducibility, the conference in September was very educational, and it really reinforced why I need to have research that's more transparent and gave me specific, even small ways to start doing that. And once I better appreciated the need for transparent research, I went from just wanting to learn enough to get by to really focus on improving my skills to the point where I no longer used SPSS for data analysis. This included learning and using different platforms for coding, building on foundational knowledge for R and Python, like learning for loops and functions, data cleaning, and code for analysis in visualizations. Lastly, I was really determined to learn more about how to create and successfully knit R notebooks. This was a critical piece for me, as my current collaborators don't use R, and I wanted to be able to share and communicate my analysis successfully with them. And because my technical skills improved, so did my confidence. Through meetings and presentations with my pod, work blocks, and a great R Squad session, I have more confidence in my ability to read, write, and troubleshoot code. And that's encouraged me to find new solutions for problems that I used to solve sort of the long way around. And with that knowledge and confidence, I was able to accomplish a few things during this fellowship. So my goal for the last 4 to 5 months has been to complete all data cleaning, analysis, and visualization for my dissertation project using R and Python instead of SPSS. To do that, and I was able to do that, I created an R notebook that's organized and easy-ish to follow and to modify as things change. It includes some skills I've improved on, such as for loops and functions. And by creating that notebook, I was able to figure out how to knit the entire analysis to create one single document that's easy to read and includes only that which is really necessary for the report. It allows me to share this analysis with my collaborators who are less familiar with working in R. And this included some amazing tables, thank you Audrey, and some fun correlation matrices, thank you to Andrew, that are clear and pretty publication ready. Not related to my primary goal though, I was also able to do a few other things thanks to ReproRehab. I created a code in R for this paper that's kind of based on this paper on how to convert TBI quality of life is a big assessment that's used in the TBI model system, and how to use those subscales to create composite scores, which is a pretty long and errorful task. But now using this code, you can use exported data straight from REDCap to convert to composite scores really easily. And I'm working on figuring out how to get this out there for others to use it as well. So all, 10 of us in the country who use the TBI quality of life scale will save hours of time. And I also gave an in-service to my PhD cohort at NYU on the principles of transparency, reproducibility, and replicability, both in quantitative and qualitative research, and that was based in part on the information I learned during my time with ReproRehab. As for planned publications, I currently have one manuscript under review where the coding skills learned here at ReproRehab were used, and so you're referenced there. And all of my future publications, may they be plentiful, from my dissertation will also credit ReproRehab. So yeah, thank you all so much for the support, collaboration, resources, and great community.